This reviews life's properties and some general concepts of life's origins and evolution. The relationships that Darwin saw in the diversity of living species led him to his theory of evolution. They also led observers to the common properties shared by all living things, and these are complexity. This means more than an intricate organization of parts. In life, the parts are constantly changing. The changes themselves are complex, highly ordered, and regulated. So complexity is therefore a dynamic organization of cellular and cellular components, the components of life. Homeostasis refers to the ability of cells and multicelled organisms to maintain stable internal environments in the face of environmental, chemical, and physical challenges. Maybe I should be clicking here. There you go. So homeostasis is a property of resistance to change. Reproduction is, of course, the one obvious prerequisite of life. Remember the statement from cell theory, cells come from pre-existing cells and not from spontaneous generation. Growth and differentiation, or development, are also obvious events. Fertilized eggs multiply and differentiate into tissues and organs. But even the lowly single-celled bacterium can grow and differentiate to a certain extent. They can become spores under adverse environmental conditions, and then can resume an active bacterial lifestyle when conditions improve. We can look at these transitions as the forerunners of the more familiar growth and differentiation I just noted. Irritability simply refers to the fact that living things are excitable. When stimulated, physically or chemically, life responds. Of course, doing anything, including living, requires energy. We will look at concepts of bioenergetics in this course. Finally, evolution is itself a property of life. All species evolved, and all living things are subject to natural selection and further evolution. So these are then the traditional properties of life as they are defined in most textbooks. Living things achieve life's properties in different ways, but underlying the apparent differences in lifestyle are a set of unique characteristics. From statements of cell theory in the 19th century, we learn, of course, that all life is cellular. From the biochemical and molecular studies of the 20th century, we learn that all life forms share the same basic biochemistry, including a common genetic code. The information necessary for life is in the sequence of nucleotides in nucleic acids, and the information flows in the same direction in all cells as shown here. This flow of information, Francis Crick called the central dogma of biology. Information flows from DNA to DNA, that's replication, from DNA to RNA, that's transcription, and from DNA, or rather RNA, to protein, that's translation. Genetic information is encoded, and the genetic code is universal, as I implied a moment ago. Here's a cartoon of two subunits of a ribosome, there it is, translating a messenger RNA into a polypeptide. This could be an E. coli bacterial or a human, say, liver ribosome. In fact, given the same messenger RNA molecule, either ribosome will make the same polypeptide, that is to say, one with the same amino acid sequence. This is what we really mean by a universal genetic code. How can we explain the fact that all cells have this universal genetic code? And also, by the way, the same basic mechanism for decoding genes, the same kind of machinery, ribosomes, messenger RNAs, and so on. Let's talk about the origins of life. All life in existence today can be traced back to a single cell, which is called the progenote. Of course, we can only imagine who and what this cell was, but we can be certain of two things. That the progenote was one of many cells and cell types that coexisted at some point billions of years ago on Earth, and that the descendants of the progenote evolved into the diverse life forms we see today. This is the best scientific explanation of how all cells and organisms come to share the same biochemical way of doing the basics of living. Let's contrast life's origins and this property of life called evolution. Understanding the former requires that we can postulate a reasonable scenario for how the physical conditions and the molecules in a primordial soup might have favored the formation of cells, that is, entities with all those properties of life. Evolution is a property that happens only to living things, after cells emerged on Earth. One of the earliest evolutionary events must have been the spread of the descendants of the progenote, at the expense of other cells. Other lineages of cells, perhaps with DNA, but with different genetic codes or different ways of harnessing environmental energy, for example, must have gone extinct very early on. Let's take a moment to understand evolution in molecular terms. This slide should be a reminder that mutations or changes in the nucleotides of DNA typically occur during replication and, of course, are random. The three possible consequences, well, here, let's take a look first at replication. Two strands of DNA in a double helix separate, new strands are made. What really happens in most rounds of replication is that some nucleotides are incorrectly incorporated in the process of synthesizing the new strands. This process of mutation is, of course, random, as I said. We're looking here at mutations that are essentially base substitutions. There are three possible consequences for the next generation during such replication. A defective or non-viable individual or cell may form. Theoretically, an individual that is better suited to survive, that is in fact more fit, might ensue. Or there might be no consequence at all. The mutation essentially is neutral. The individual organism might be unaffected. So, the concept of fitness applies to individuals and not to species. Let us consider these fitness consequences in more detail. Defective individuals, those with deleterious mutations, won't survive, or at least won't be able to reproduce as well as the rest of the population. Individuals with beneficial mutations would be selected immediately because they would be presumably stronger, they could live longer, they could reproduce longer, and have more offspring. But actually this sort of thing is pretty rare. More likely, most mutations in organisms that survive are initially neutral, with no effect on individual fitness. The individual spreads these kinds of mutations by reproducing, but the progeny are no more fit than the parent. A neutral mutation might be recognized as useful much later, when environmental conditions change, we would say to prepare, or rather to favor the prepared genome. Evolution leads inevitably to speciation, that is to say, the formation of new species. At some point, the accumulation of naturally selected traits leads to physical or physiological differences in descendant populations that, perhaps along with physical separation, make it impossible for them to mate with other lineages from the same set of forebears. In other words, speciation occurs when populations of individuals become reproductively incompatible. The diverse organisms alive today share a common heritage of genes and proteins, many of whose nucleotide amino acid sequences can be compared and traced backwards in evolution to reveal a history that is remarkably similar and even more accurate than the first evolutionary trees that the older biological generation generated based on morphological similarities between organisms. In this example, it may look familiar to some of you, ribosomal RNAs that are part of the structure of the small ribosomal subunit in different species were compared. We can see a few base substitutions found in different species in the highlighted part of the sequence. Here's another version of the sequence that was originally up here. Here's yet another, and finally a third one. Note, by the way, that this RNA molecule is shown highly folded due to internal hydrogen bonding. When these small ribosomal subunit rRNAs or ribosomal RNAs from many different species are compared, it's possible to group the species by how close their rRNA sequences are. This is a molecular taxonomy. By this analysis, all species in the world today can be grouped into three major domains: the bacteria, the archaea, and the eukarya. Using computers to pick the shortest possible pathways by which base differences between these species, between the RNA in these species, could have occurred, that is to say, pathways which involve the least number of base changes to get from one species to the next, it's actually possible to infer a common ancestry of related species, and ultimately of all existing species. One can even infer a reasonable base sequence for the small ribosomal RNA subunit for each of the ancestral species, including the progenote down at the bottom. If the entire informational content of a species is defined as its genome, then what we can say is that evolution remodels genomes, reshapes them, changes them, in other words, and quite dramatically at times. The Human Genome Project was completed in 2003, only 13 years after it was begun. The many different genome projects completed since are revealing just how evolution is remodeling genomes. Between species, we see not only base substitutions, but differences in patterns of gene structure, that is, for example, introns and no introns, genes with introns, genes without introns. We see differences in patterns of gene organization. You may recall that bacteria have genes organized into operons, and eukaryotic cells do not have operons. We see changes or differences between species in how older genes are co-opted to have new functions. That is, new functions are either added to existing genes or genes are turned entirely over to new functions. We also see changes in evolution in the timing of the regulation of gene expression. In other words, we sometimes have to broaden our definition of mutation to explain evolution and the diversity of species at the molecular level. I'm one slide behind, but basically this is what I've just spoken about. And that brings us to the end of this presentation.